On today, let us see about the animal cell culture, how to culture animal cells and where the animal cell culture techniques developed. So, more things we shall discuss in this video. So, history of animal cell culture. It was the first person, Jolly, who in 1903 showed for the first time that the cells can survive in vitro. Then the persons like Ross Horizon, Alexis Carrel, Enders, Weller, Robbins. The Robbins grow the poliomyelitis viruses in culture. So accordingly, the techniques in the animal cell culture developed. So first now let us see about the salient features of this animal cell culture. So these animal cells can grow in simple glass or plastic containers, okay, with nutrient medium. But they can grow only up to limited generation. Like microorganism, we cannot culture these animal cells very easily. It's somewhat difficult when you compare to microbial culture. And also these animal cell exhibit contact inhibition in culture. So the cancer cells are apparently different from the normal cells. Most probably tumor cells are preferred for experimental purposes because these tumor cells multiply fastly. And also there is a difference in the in vitro and in vivo growth pattern of cells. For example, if there is a cell to cell interaction and cell to matrix interaction, uh, you can see um, that is if there is a cell to cell interaction and cell to cell matrix interaction, that is normal development of cells. But if it is not there, then you can go for the secondary cell culture, okay. And also if there is a lack of three-dimensional architectural appearance, that is animal cells should have the 3D appearance, okay, architecture. And also change hormonal or nutrition development. That is the way of adherence to glass or plastic container in which they grow. Some cell proliferation and cell shape of shell results. Some alterations will be there. As you know, from in animals, it will be in one shape, but the same cells when you grow in artificial conditions, some differences you can see. So, and also the maintenance of growth of cells under laboratory condition in suitable culture medium is known as primary cell culture. That is up to a limit only primary cell culture can be done. After that, you should go for the secondary cell cultures. In the primary cell culture, some of the shape, size, etc., the characteristics of the animal cells can be maintained. But when you go for the secondary cell culture, some differences you can see. And also cells are dissociate, how you are going to dissociate the cells from the tissues by means of enzymatic digestion or by means of some other digestion, mechanical digestion methods. You can detach the cells and you can go for your experimental procedures. The primary culture is subcultured in fresh media to establish a secondary cell culture, okay. The various cell types can be distinguished as finite cell line and continuous cell line. Finite cell line have a limited lifespan, that is normal cell culture. It has only a limited lifespan and also it cannot multiply very fastly. It can grow up to a limited limit only, and also a limited number of generations can only be produced. Normally, it can divide 20 to 100 times. 100 population, 20 to 100 population doublings before extinction. But when you take for tumor cells, that is, it can give numerous cells. So that only in experiment procedures, numerous tumor cells are being preferred by the researchers. So the continuous cell lines are transformed, immortal, and also tumorogenic. So that is only that is about the continuous cell lines. And what are the environment conditions you have to give for the growth of animal cell culture is the physical environment is very important. You should provide optimum temperature, optimum pH, osmolality has to be under control, gaseous environment has to be checked. Supporting surfaces, animal cell culture usually grow when there is a supporting surface only. The cells from chemical, physical, and it should be free from chemical, physical, and mechanical stress. And the nutrient media that you are going to use for the animal cell culture has also been checked, and it should be in definite medium. And serum is popularly used for animal cell culture. Serum free media is also available. Serum culture has some growth factors for the development of animal cell culture. Cryopreservation methodology you can use, that is for storing the cells. After culturing the cells, if you want to store the animal cells, this by method of cryopreservation you can store the cells at very low temperature, at that is minus 180 to 196 degrees Celsius. You can store the cells by using cryopreservation technology and liquid nitrogen, that is DMSO, liquid nitrogen. Okay. And how will you maintain the aseptic condition? All the things you have to sterilize. Everything has to be sterilized. And also carbon dioxide in incubators, inverted microscope, low speed centrifuges. These are some of the instruments that has to be used in an animal cell culture laboratory. And what is the difference between organ culture and histotypic culture? Organ culture, here you can see the native tissue that retains most of the in vivo histological features. That is from the parent cell. That is from the parent cell means from the cell where you are going to take from, it's from an animal or human being. From that you are taking the cell and going to culture in the laboratory, and you can see most of the characteristics remain same as the parent cell culture, that is organ culture. But histotypic culture is nothing, is not like that. You can see the re-aggregation to form a tissue like structure represents histotypic culture. Okay, some differences you can see in the histotypic culture. Next, we can see about organotypic culture. There are three types of culture, that is organ culture, histotypic culture, organotypic culture. This culture technique involves a recombination of different cell types. Okay, recombination of different cell types to form a one or more different tissues or organ. Uh, so this is organotypic culture. Here you can see the cell lines. Cell line means the particular cell lines that you are getting from a particular animal cell culture. Okay. So there are some terms you have to see. The first one is split ratio. This is nothing but the divisor of the dilution ratio of a cell culture at subculture. Okay. Passage number, it is the number of times that the culture has been cultured. And the passage number, you should be very careful. That is the number of times the culture has been cultured. Okay. Then generation number. It refers to the number of doublings that the cell population has undergone. That is the number of doublings, how much times the particular cell has been divided. Okay. So these are the terms you have to be thorough when you are studying the animal cell culture. And requirements for an animal cell culture, as you know, temperature is very important, and normal temperature you have to maintain is 37 degrees Celsius. And culture media, the culture media can be prepared in many ways, but we should be careful with the nutrient content. Okay. And also pH, osmotic pressure, etc. should be very carefully maintained. The chemical constituents while preparing the culture media also is very important. The presence of inorganic salts, nutrients should be taken into careful account. Okay. That adherences, as you animal cell culture can be maximum you can culture animal cell culture using an adherence safe only, because the cells will adhere to the particular surfaces, there it will multiply, and you can then harvest the cells. Suspension cells can also be used. This is, for example, blood cells, that is hematopoietic cells, etc. Malignant tumor cells can be cultured using the suspension cell culture methodology. Okay. Primary culture, as we discussed, it can be done only up to a limit. The primary cell culture will take its characteristics from its parent cell only up to a limit. After that, it has to be separate. It will go for a, we should go for a secondary cell culture. Okay. Here, this the same thing. Finite cell culture. Finite cell culture, up to a limit only it will divide. That is, finite cell cultures are formed after the first subculturing or passaging of a primary cell culture. These cultures will proliferate for a limited number of cell divisions, after which they will senescence, that is died. The proliferative potential of some human finite cell cultures can be extended by introduction of viral transforming genes, SV40, transforming antigens. So, by transforming the viral gene, you can extend the a life of the particular cell. Okay, so that is finite cell culture. Continuous cell lines, you can go, you can do it for number of numerous times. You can go for the culture. That is continuous cell lines, and you can get the immortalized cell using this continuous cell culture. Okay. Uh, then culture conditions, that that is the how much amount of pH normally 7.4 we will maintain, 37 degrees Celsius will be given, and also carbon dioxide incubator can be used. uh, pH has to be very carefully noted while you are doing the animal cell culture. uh, so so uh, the following protocol can be used to uh, that is to culture the animal cells. It may be anchorage dependent or anchorage independent. How you will get the tissues from the particular animal or human cells by trypsinization method. By mechanical destruction method also you can get the animal cell culture. By enzymatic methods like trypsination, by that methodology also you can get the animal cells from the particular animal or tissue, and you can go for the animal cell culture. Okay, so inoculate cell grown in serum based medium intermediate containing, that is how much percentage of serum you have to take. Then subculture after 48 hour, growth for 2 passages, increase the content of serum free medium. If you want to for more, you can repeat the steps. Okay, so that is given here. Here by how trypsin methodology you can take the animal cells. Here you have to remove the media, the flash stand for 30 seconds. This is just how the conditions has to be maintained to get the animal cells from the particular animal. Okay. Then all the things you are going to use for the animal cell culture has to be sterilized. Whether it may be a media, or it is a flask, or it is a pipette, or it is a test tube, or it is a measuring cylinder, anything else you are going to use for the particular animal cell culture has to be thoroughly sterilized. 121 degrees Celsius for 15 minutes is very compulsory. You have to follow while doing all the animal cell culture. Uh, then you have first step, it's a sterilization only. You can see here preparation and sterilization of the substrate, that is culture vessels, preparation and sterilization of the medium. All the mediums you are going to use for the particular cell culture, you have to sterilize it. Isolation of explant. Explant is nothing but your it may be a tissue or animal cells where you are going to take that thing. Disaggregation of the explant. The outer part has to be removed, that is sterilized. For sterilization process only, you are going to remove the outer part, taking the inner part. The cells has to be trypsinized or mechanical destruction. You can go for separation. Anything else, you just prepare, separate it, and then go for subculture cloning, and without contamination you can get the product. So that is autoclave, as you all know, as you have studied in microbiology. Autoclave can be used for the sterilization of the things you are going to use for animal cell culture. Okay. Isolation of explants. Explants can be dissected out following an appropriate protocol developed to avoid contamination and retain the cell survival. Cell survival is very important. Okay. In general, embryos and other content of uterus are free from contamination. Okay, but in adult tissues, bacterial contamination may be common. In simple terms, the site of opening is sterilized. So, where you are opening in an animal, the site of opening has to be sterilized with 70% alcohol. The tissues are removed aseptically and usually placed in a balanced salt solution, that is BSS solution, okay, or HBSS solution, highly balanced salt solution, supplemented with antibiotics, any antibiotics. All subsequent handling should be under strictly aseptic conditions, preferably under laminar flow cabinet. All the things has to be practiced under laminar flow cabinet or hoods or in sterile rooms. Care should be taken not to violate ethical or legal boundaries in obtaining the desired explant. Okay, while doing animal cell culture, the ethical and legal issues you have to thoroughly follow, because it's not like a microbial culture. Animal cell culture is different from microbial cell culture. Here, all the ethical issues has to be taken into account. And disaggregation of explant. Usually, how you will disaggregate the explant cell cultures uh, are generally started from disaggregated explants. Occasionally, cells do migrate out of cultured whole tissues. Okay, in such cases, the such cells are then separated by trypsin. Trypsin you can use or other enzymatic treatment to establish cell cultures. But in general, the tissues are first disaggregated in one of the following ways. What are the ways? Three ways: mechanical disaggregation, enzymatic disaggregation, EDTA treatment. Okay. So first, mechanical disaggregation. Here, the tissue it will be chopped into small pieces. So you have to chop the tissues into small pieces. It may be 1 millimeter cube in size. Then they are cultured in suitable vessels. The pieces are then attached to the substrate either due to their own adhesiveness, or the dish may be scratched. Okay, you can scratch the dish to facilitate attachment, or you can use plasma may be used. Cells grows out of the tissue pieces were trypsinized and subcultured. The explants themselves can be retained in the same dish or transferred to a new vessel to obtain further cell growth. Okay, so this is the mechanical method. The second method, enzymatic disaggregation, you can use trypsin. Okay, trypsin 0.25 percentage crude or pure collagenase, and collagenase, that is trypsin and collagenase, you can use as a more commonly used enzymes for enzymatic disaggregation method. Some enzymes like elastase, mucase, papain can also be used. Okay. Uh, in some things you can see it may be that the trypsin may be damaging one, that is epithelial cells cannot be much effectively removed using this trypsin, and also ineffective in the case of fibrous tissue. You cannot use this trypsin. There you have to use mechanical disaggregation method. Okay. And then EDTA treatment. EDTA treatment, you know, calcium and magnesium ions. That ions can be used along with balanced salt solution, that is BSS. You can use and get the that is you have to prepare the EDTA in balanced salt solution. Then it can be used for the separation of tissues. So these are all about the animal cell culture and how the explants can be removed from the particular tissues. Hope you all enjoy the video. Kindly like, comment, share and subscribe the video. Thank you.